Hello everyone. I am with the topic for the next digestive system, human digestive system. We have already studied, and the remaining topic, the mechanism of digestion, we have to discuss. So the topic today is digestion in human, and I have already explained that digestion in human will be divided into two parts. The first part is alimentary canal, and the second part is digestive gland. These two parts here we have to study, and their functions as well. So these parts and the functions will be very important, and by these explanations only we can understand the complete process of digestion. For this we need to draw again human digestive system, and where I will explain the complete process and mechanism of digestion. So let me draw the diagram of human digestive system. This is the part I am drawing here of alimentary canal is the large intestine, and the end of this is the bag-like structure here. Just below the stomach, this is the stomach, just below of this a leafy structure will be seen. This leafy structure is called pancreas. A liver is present just behind the stomach and attached with the food pipe. For our convenience, I need to sketch the facial part of human being also, so that we can understand the remaining part of. So this is human digestive system. Actually, we can say alimentary canal plus glands, and small intestine is all around here. The size of small intestine is bigger than the large intestine. So there will be a question, why small intestine is called large intestine? Why the small intestine is larger in size? The reason is this small intestine is very long, almost 6.5 meter in the length, but the large intestine, it is around, this is comparable, very short. As you can see here, it is in the diagram, the size is very bigger of small intestine. Why it is called small intestine? The reason is this is very broader in appearance. The broad structure of large intestine is here, and that is why this is called large intestine and this one is small intestine. So the food is entering into the mouth. This is the mouth. Through mouth the food has been taken, and a very important board examination question is going to solve here. In what part of human alimentary canal? Alimentary canal is a part which starts from mouth and end to the anus. From mouth to anus, the tubular structure, this is called alimentary canal. And this alimentary canal is consist of mouth, esophagus. This esophagus is also called food pipe. This is called stomach. This is duodenum. This is duodenum. This is a small intestine. This is large intestine. As I have already told you, the undigested part will be stored temporarily into the rectum, and as soon as the size will increase, the information will go to the CNS, central nervous system, and we will feel the toilet, means we need to go to toilet, and it opens through this anus. Sphincter muscles are there in rectum. Rectum is made up of a special muscle which is called sphincter muscle. That sphincter muscle helps the undigested food particle to get out of the anus, means it will not go out of the anus itself unless the CNS will not work. So this is a complete structure of human digestive system made up of alimentary canal. So alimentary canal name I have made with the blue color. Labeling is made by blue color. This red color labeling is of the gland. Liver gland, the most largest gland of human body is liver. This is pancreas. Another gland is there into mouth cavity, which is called parotid gland, and for your convenience I can write salivary gland. So these are the glands, associated glands for the human digestion. So what will be the process here we have to discuss. Remember, whenever you are drawing any diagram, the concern means the name of the concerned diagram to be mentioned just below the diagram. Like I have drawn here human digestive system, so here I have to write, this is very important thing to understand, human digestive system should be written, no, must be written. So I have mentioned here human digestive system. Likewise, you have to also do it in the examination, in fact everywhere when you are going to draw any diagram. So what will be happen if we are going to perform digestion, our body is going to perform digestion. That's a very easy and simple process. You take food, the food goes into the mouth, the process is called ingestion, and after that digestion will be started. And the most important question which I started, that which part of alimentary canal, from mouth to anus, the part is called alimentary canal, which part of alimentary canal digestion starts? Usually people understand that it starts from stomach. No, this is wrong answer. The correct answer is digestion, it starts from mouth only. So as soon as we take food, the food goes into the mouth. We chew the food by using teeth. The parts of teeth I have explained already. The teeth help the food to make it paste. That paste is having a gland inside the mouth cavity. A gland is there which act on the paste like the food is made. So let me clear. The first gland is working, which is salivary gland. How it is acting? Salivary gland that secretes saliva. Salivary gland secretes saliva, and this saliva has an enzyme. That enzyme is called salivary amylase. And that salivary amylase, what does it do? The starch present in the food, will be broken down in presence of salivary amylase into maltose. So the breakdown of starch, it starts from the action of salivary amylase, which is secreted from salivary gland, and salivary gland is present in the mouth cavity. So this is the very first step of digestion, which is starts itself from the mouth cavity of human being. So the answer will be mouth is a part of alimentary canal from where digestion starts, because the starch present in the food will be digested into maltose by the action of salivary amylase secreted by salivary gland. So this is the very first action taken into the mouth. Now the food has to go to the stomach by a specific movement. I have explained it already, which is called peristaltic movement, or this is also called peristalsis. So with the help of peristalsis, food goes down towards the stomach. Now stomach secretes three types of secretions that secrete gastrointestinal juice. gastrointestinal juice will be secreted, which has three parts. These three parts are, the first is HCl, the second thing is pepsin, and the third substance is produced is mucus. Remember, three things are secreted by stomach, that is gastrointestinal juice. Three things. The first is HCl. The food we are taking, we are not going to check it with the help of lens. We are taking a bite of food, and before putting it into the mouth, we check it with the lens. It never happens, even it is not possible. So we take it, and some microorganisms may enter into the alimentary canal. So that those microbes which can severely harm us, and that's why they will be killed by the action of HCl released by the stomach wall gastrointestinal juice. So this HCl will be released. HCl kills let me write here, kill germs present in the food. Pepsin has very important function. This pepsin what does it do? It acts upon, let me write here, protein. This protein will be broken down into amino acids. The breakdown of protein or digestion of protein will be done by by action of pepsin. So this pepsin digests the protein into amino acids, right? So these amino acids will be produced. These amino acids, never forget, this complete digestion of protein will not be occur here, and likewise the complete digestion of the starch is also not happened here. So no starch complete digestion will be done here, means some of the starch will remain left. It has to be digested completely, one of the part of alimentary canal in the small intestine, I will tell you later, and some of the part of protein will remain left even after the action of the pepsin. So some of the protein will be digested here in the stomach. Now the important function of this mucus, so many times the most important part of this is function of mucus. There will be so many times question in the board examination, what is the role of mucus in the digestion? So the HCl, hydrochloric acid, is present in the stomach. It can kill the microbes present in the food, but it can harm also if it touch with the wall of stomach. So stomach wall can be harmed by the action of HCl. Therefore, a protective layer is present. The protective layer I have made with the red color marker so that you can understand. The mucus is red colored marker, I have made it, and action of HCl will not be occur on the stomach wall, and your stomach wall will remain protected from the action of HCl. So this mucus layer protect the stomach wall from the action of HCl. This is the one of the most important part of the secretion. So up to here the digestion has been completed. Now the next part of the alimentary canal is opening of stomach, and the very first part of small intestine is duodenum. So this duodenum, what does it receives? Let me show you. There is bile juice secreted by liver. This bile juice will be stored into gallbladder. This is called gallbladder. This gallbladder release bile. The bile is coming in duodenum. Pancreas release pancreatic juice. Another gland, the gland salivary amylase, salivary gland is done. Liver secretes bile which is stored into the gallbladder, and now pancreas is secreting three types of secretion. So gallbladder, what is the role of gallbladder? Another important question. Gallbladder release bile which never take part in the digestion of food. So where does it take place? So it takes place in the conversion of medium of food, whatever food we are taking, action of acid will be also done and the medium of food become acidic. So to neutralize the acidic medium of the food, the basic substance will be secreted by the liver and which stores into the gallbladder, which is called bile. That bile convert the medium of food from acidic to basic medium. So the food become basic medium by the action of bile stored into the gallbladder. So gallbladder just store the bile and nothing else. So bile is not playing any role to digest the food, but it convert the medium of food so that the food will be digest easily in the next part of alimentary canal. So the food, now it was acidic earlier, now after the action of bile it become basic. The action of pancreas will be taking place. Three types of secretions will be done by pancreas, and those three secretions we can remember by our Honorable former Prime Minister of India, Sir Atal Bihari Vajpayee. The name of the former Prime Minister is Atal, so we can write ATL. This A for amylase. This T for trypsin. This L for lipase. These three secretions are responsible for the further digestion of food. These three secretions will be done by pancreas, so we can call it pancreatic amylase. Amylase was once it was secreted from saliva, so it was called salivary amylase. Pancreas is also secreting amylase, so it is called pancreatic amylase. This will be called pancreatic trypsin. This will be called pancreatic lipase. So there are three types of secretions by the pancreatic juice. This pancreatic juice again joined to the duodenum where the food converted its medium by the action of bile. So it become alkaline. Now in that alkaline medium, these actions will be done. How it will be done? Let me show you. Amylase was working for starch digestion. So we will write again starch. This is the activity of pancreas. Right? So pancreas secretes, first secretion is starch converted into the same one, maltose. The same one, maltose, by the action of pancreatic amylase. Here we can write pancreatic amylase. The second is trypsin. Trypsin get converted into, trypsin. Trypsin is an action enzyme. So protein will be digested by trypsin into amino acids. AA for amino acids. And here we can write trypsin. And the third one is lipase. So we will write another arrow and here will be lipase. So this will be lipase, and breakdown of fat into fatty acid plus glycerol will be done by the lipase. So pancreatic juice secretes three enzymes. Those three enzymes are amylase, which convert the starch into maltose. Second, trypsin, which break down the protein into amino acid. And third is lipase, which breaks the fat into fatty acid plus glycerol. And here the complete digestion will be done. The complete digestion will take place into a small intestine. So what will be the next step? Absorption. So absorption, how the absorption will be possible? The absorption, the food particles now very fine in structure, therefore it can absorb by the small intestine, as small intestine wall is convenience for the absorption. Why it is convenience? This is also board examination question. Board ask what are the What are the reason behind absorption takes place in small intestine? Why? Answer will be the wall of small intestine is very fine, therefore food particle can enter. And another very important reason is this: if we cut this much part of small intestine, we will see, suppose this is small intestine, so it has finger-like projections. These projections are called villi. These villi, these are called villi, which increases the absorption surface area. Actually, surface area, the more surface area will increase the more absorption rate. So absorption will be higher because of more villi is there. Villi will be more, which increases the surface area, and the more surface area will increase the absorption rate. Therefore villi is helping for this, and that's why absorption. And it is also, the small intestine is also perforated with thousands of capillaries which absorb the food easily, and the food can enter into the blood. The blood will now transport the food, digested food, to each and every cells, and whatever the function will be there, we'll discuss it later. The small intestine, now function of the small intestine will be over. The food will finally go to the large intestine where reabsorption takes place. So we can write here, we can write large intestine. Whatever food which was not digested, what will be happen with that? The reabsorption will be occurring. And this is also a board examination question. What is the role of large intestine in digestion? We will write reabsorption of water and salt. Why so? Why it is called reabsorption? Because absorption was done once in the small intestine, and again it will be happen in the large intestine, and therefore this is called reabsorption. And reabsorption of only two things will be done: salt and water. Salt and water will be reabsorbed into the small intestine, and what will be happen actually? The thing will be happen, the water and salt. These both, salt and water, will be absorbed from the food, and that's why the stool, the feces, the toilet material will be solid. And this is also a board examination question. In boards there will be a question, why the stool, the fecal material is called stool. Why the stool is solid? Because reabsorption is done in the large intestine of salt, of that food, undigested food, salt and water. So if water is taken from the any mixture, any solution, that becomes dry and it becomes solid. Therefore the undigested food which we remove every day in the morning is always solid. So this is all about the storage, large intestine function. Rectum is a place where the storage will be done, and as soon as it become large, the food will be the undigested food will be removed out of the this alimentary canal by using anus, and this will be controlled by a muscle. And this is also an important question. That muscle is called sphincter muscle. This muscle is called sphincter muscle, which control the food, undigested food, removal of that undigested food material from rectum and from outside of the body through the anus. So this is all about the digestion part. Let me explain it in Hindi. I will try to short it. ह्यूमन डाइजेस्टिव सिस्टम दो पार्ट से बना होता है, एक है एलिमेंट्री कनाल और एक डाइजेस्टिव सिस्टम। ये एलिमेंट्री कनाल है जो माउथ से लेके और एनस तक फैला होता है। इस सिस्टम में फूड जैसे माउथ के रास्ते अंदर जाता है, एक एलिमेंट्री कनाल होता है, ये माउथ से स्टार्ट होता है, जहां पे सैलिवरी ग्लैंड होता है। वो ग्लैंड्स तीन होते हैं हम लोगों के बॉडी में, जिन तीन ग्लैंड्स को तीन रेड कलर से हमने लेबल किया है। पहला है सैलिवरी ग्लैंड, दूसरा है पैंक्रियाज और तीसरा है लीवर। ये तीन ग्लैंड्स हैं, ये तीनों ग्लैंड्स और एलिमेंट्री कनाल मिलकर ही ह्यूमन डाइजेस्टिव सिस्टम बनाते हैं। सैलिवरी ग्लैंड ने क्या किया? सलाइवा सेक्रेट किया, जिस सलाइवा में सैलिवरी एमाइलेज होता है, और वो सैलिवरी एमाइलेज स्टार्च को माल्टोज में कन्वर्ट करता है। मतलब डाइजेशन माउथ से ही स्टार्ट हो गया, और यही आंसर होगा बोर्ड में अगर क्वेश्चन आता है तो, कि डाइजेशन माउथ से ही स्टार्ट हो जाता है। फिर उसके बाद से फूड आता है स्टमक में, एक पेरिस्टाल्टिक मूवमेंट होता है फूड पाइप में, और यहाँ पे आता है जहां पे तीन तरह के सेक्रेशन होते हैं। एक HCl होता है जो किल करता है माइक्रोब्स को, दूसरा होता है पेप्सिन जो ब्रेक डाउन करता है प्रोटीन को अमीनो एसिड में, और तीसरा होता है म्यूकस। म्यूकस क्या करता है कि HCl जो एसिड है उस एसिड का एक्शन इसके इस वॉल पे ना हो, क्योंकि वॉल अगर डैमेज हो गया तो वॉल जैसे डैमेज होगा, एक्शन होगा एसिड का, तो ये बर्न हो जाएगा, और वॉल जब बर्न होगा तो यहाँ से ब्लड बाहर आ सकता है और एक डिजीज होता है जिसको पेप्टिक अल्सर कहते हैं। तो वो पेप्टिक अल्सर नहीं होगा क्योंकि यहाँ पे म्यूकस होता है। तो ये 3 एक्शंस हो गए। फिर फूड आता है डुओडेनम में, जो स्मॉल इंटेस्टाइन का पहला पार्ट होता है और आखिरी पार्ट होता है स्टमक का। तो ये क्या करता है, यहाँ गॉल ब्लैडर से बाइल को रिसीव करता है। गॉल ब्लैडर का फंक्शन होता है बाइल को जस्ट स्टोर करना, और वो सेक्रेट होता है लीवर से। तो जो बाइल यहाँ प्रोड्यूस हुआ है वो बाइल डाइजेशन में हेल्प नहीं करता। करता क्या है, वो फूड को, उसके मीडियम को चेंज कर देता है। एसिडिक फूड मीडियम होता है जिसको बेसिक में कन्वर्ट कर देता है ताकि बाकी एंजाइम के एक्शंस हो सके। एसिडिक मीडियम में फूड में एंजाइम एक्शन नहीं होता है। तो ये पैंक्रियाज क्या करेगा, पैंक्रियाज फूड को 3 सेक्रेशन करके और डाइजेस्ट करता है। वो 3 सेक्रेशंस हैं। इसको मैंने एक ये दिया है, निमोनिक्स दिया है, जिससे आपको आसानी होगी इसको याद करने में। फॉर्मर प्रेसिडेंट, जो ऑनरेबल प्रेसिडेंट है हम लोगों के, अटल बिहारी वाजपेयी जी, उनके नाम से अटल, ATL, A फॉर एमाइलेज, पैंक्रियाटिक एमाइलेज, और वो क्या करता है, स्टार्च को माल्टोज में कन्वर्ट करता है। T फॉर ट्रिप्सिन, ये T है, ट्रिप्सिन, ये ट्रिप्सिन क्या करता है, प्रोटीन को एमिनो एसिड में कन्वर्ट करता है। और लाइपेज, L फॉर लाइपेज, जो फैट को फैटी एसिड और ग्लिसरॉल में कन्वर्ट करने का काम करता है। उसके बाद फूड आता है स्मॉल इंटेस्टाइन में। तो ऐसा माना जाता है कि स्मॉल इंटेस्टाइन में डाइजेशन कंप्लीट हो जाता है और स्मॉल इंटेस्टाइन में एब्जॉर्प्शन होता है। अब एक क्वेश्चन और होता है बोर्ड का कि स्मॉल इंटेस्टाइन में ही एब्जॉर्प्शन क्यों होता है, क्योंकि स्मॉल इंटेस्टाइन में 2-3 कंडीशंस होते हैं। उसमें विलाई होता है जो सरफेस एरिया को इंक्रीज कर देता है, जिसकी वजह से एब्जॉर्प्शन ज्यादा होता है। फिर दूसरा रीजन होता है कि उसमें बहुत ढेर सारे ब्लड कैपिलरीज होते हैं जिसमें ब्लड में इजिली वो एंटर कर जाता है डाइजेस्टेड फूड। और तीसरा होता है कि उसमें जो स्मॉल इंटेस्टाइन के जो वॉल होते हैं वो बहुत ही थिन होते हैं, जिसकी वजह से फूड इजिली एब्जॉर्ब कर लेता है। फिर फूड आता है, वहां से एब्जॉर्प्शन के बाद वो पूरे सेल में डिस्ट्रीब्यूट हो जाएगा। अब वो फूड जो डाइजेस्टेड नहीं हुआ, एब्जॉर्ब नहीं हुआ, वो आ गया लार्ज इंटेस्टाइन में। लार्ज इंटेस्टाइन उस फूड को क्या करता है, उसमें से रीएब्जॉर्प्शन करता है। एब्जॉर्प्शन तो हो गया था, फिर से कर रहा है, इसीलिए इसको रीएब्जॉर्प्शन कहते हैं। रीएब्जॉर्प्शन किसका करता है, साल्ट और वाटर का। साल्ट और वाटर का रीएब्जॉर्प्शन होगा, और इसी वजह से जो टॉयलेट हम रिलीज करते हैं मॉर्निंग में, वो स्टूल जो नाइट्रोजनस वेस्ट रिलीज करते हैं हम, वो जो होता है वो सॉलिड होता है। सॉलिड इसलिए होता है क्योंकि उसमें से वाटर रीएब्जॉर्ब हो गया और इसीलिए वो सॉलिड हो जाता है। फिर वो आता है रेक्टम में, स्टोर होता है, और स्टोर होने के बाद एनस से बाहर हो जाता है। तो एनस से बाहर करने के लिए एक मसल्स है जो हेल्प करता है, और वो होता है स्फिंक्टर मसल। सो स्टे विद मी एंड एंजॉय योर फर्दर क्लासेस फॉर 10th स्टैंडर्ड। थैंक यू, हैव अ नाइस डे।